Thank you, everyone, for coming, and thank you to the organizers. So today I'm going to be talking about trees and developmental biology. And I've posed a question here, but I won't give too many answers. So this is very preliminary work, but hopefully I'll throw out some of the main ideas and some of the things we've worked on so far. So I'm a postdoc in Tanya Stother's group at ETH. And this is work that I've been doing over the past year or so. Okay, so what do I mean when I say trees in developmental biology? So these are single cell level trees. So here, for example, we start from a progenitor cell, so an egg cell in this case. This is a very ambitious figure. And each internal node in our tree is a single ancestral cell. And at the tips, we have a sample of individual cells. So the goal is to take a sample of cells from a single time point, relate them all back to a single progenitor cell. And we are primarily concerned with healthy development here, so healthy tissue and kind of normal healthy development. And the idea is that if we can better understand healthy development, then perhaps we can also eventually better understand developmental diseases. But throughout this talk, I will be focused on healthy development. Okay, so why do we need the lineage tree? So the ultimate goal is to understand developmental trajectories. But in order to do this, we have to consider that these measurements are correlated to the lineage. So for example, the transcriptome can be measured at a single cell resolution, but what we have is we have a sample of cells sequenced at a single time point. And to accurately analyze these, we have to take into account correlations that can arise due to the lineage. So a certain type of experiment leverages the reproducibility of development by taking, instead of allowing cells to develop for a certain time, sampling, and then sampling at multiple time points. But each of these samples have their own lineage. So again, even with these types of time course data, we still need to take into account the cell lineage. So these developmental trajectories we can't directly observe, but they occur kind of along this lineage. And one of our first goals with this project is to understand how accurately we can infer the lineages from the data that we have. So what data do we have? I'm going to focus here on high throughput sequencing methods. So the first thing that might come to your mind is genomic data. The technology does exist. We have single cell DNA sequencing. But again, I'm focused on healthy development, and we really don't expect many mutations to arise in the DNA here. So while, yes, we can get this data, we do not expect it in this case to have any phylogenetic signal. What about epigenetic data? So for example, DNA methylation. This sequencing technology is emerging. So this technology is kind of in a very developing stage. But these data sets are not very common. So we don't know if there's phylogenetic signal. We also don't have a ton of data. So while this may be promising in the future, this is not what I'm going to focus on here. Okay, what about transcriptomic data? So we can get a measurement of all of the transcripts in this cell. And this technology is well developed. These data sets are very commonplace. So this is kind of, one of the main sources of data that we have to work with. But understanding its phylogenetic signal has been much less studied. There is some assumptions that are currently made that there must be phylogenetic signal in this data, but this has currently not been, to my knowledge, well validated, and that was one of the goals that we want to do here. I also want to introduce lineage tracing data because this is going to be our source of ground truth data that we have to compare against. So these technologies have been developed for quite some time now. So for example, here I'm showing an example of a static barcode where cells have been equipped with a marker, so a fluorescent marker in this case, which allows us to resolve kind of broad scale lineages at a later time point. So this is just one example of kind of an early barcoding technology. I have an asterisk next to the yes here, because while these data do exist, they're not super commonplace. There's not many labs that are able to actually generate these data, although it does have very high quality phylogenetic signal. And this is typically going to be our ground truth data in this field. So the first thing I want to talk about is a project that we did on lineage tracing data. So understanding when we do get accurate reconstruction from these data, so that if we can understand this, then we can move on to the more difficult picture, I suppose. So before I introduce the static barcode, we also have dynamic lineage tracers. So for example here, this is kind of an early generation dynamic lineage tracing technology. So most of these are based on CRISPR arrays. So here we have an initial cell, which is equipped with a kind of empty CRISPR array. And then as the cells develop and divide, these arrays accumulate edits dynamically. And so in this way, we were able to ideally resolve smaller scale lineages, as opposed to in the static case. A few things to point out here is that, yeah, the edits are heritable and they're also irreversible. So this is a really nice property of this kind of CRISPR machinery, is that once a site has been edited, it cannot be targeted again. We can have things go wrong, we can have masking or dropouts, but we do have this really nice property of irreversibility. Another nice thing about these kind of engineered constructs is that they're very tunable. And this is where we felt that modeling could be a useful approach here, because we can start to then dig into these tunable parameters and understand where we can sort of direct the technological advancements in this field. So without getting into too much detail, we investigated the theoretical accuracy of tree reconstruction from a particular class of dynamic lineage tracing, dynamic lineage recorders. So this is just an example showing parameter regimes or experimental setups where we do get very accurate trees with high probability. So if you're interested in that, I could talk about it more. But the goal of this is just to say that we can have ground truth lineage trees in this field, which is exciting because then we can ask our next question, which is, what about transcriptomic data? So given that we have this high quality lineage tracing data, what's left to do? Like, why I'm asking this question? And I alluded to this before. The problem is that these lineage tracing experiments are very expensive. They're very technologically demanding. Not a lot of labs can do this, and not a lot of labs routinely produce this data. Single cell RNA-seq is, by contrast, very commonplace, and there's a lot of these data sets around. I mean, in an ideal scenario, we do have both sources of data. So we have transcriptomic data together with kind of high quality lineage tracing data, and then we can analyze these in a combined framework. But these kind of combined data sets, unfortunately, do not really exist, except for very few examples. So yeah, there is this kind of push in this field towards a natural lineage. These lineage tracing experiments are also very invasive, so there's some model systems that we can't even perform these on. Okay, so yeah, in most cells, there's only a subset of genes that are active at any given time. So these will be the cells that are relevant for its current developmental stage and potentially like housekeeping genes and maybe some others, for example. Transcription is regulated by epigenetic mechanisms, and most of these are thought to be heritable. So that's kind of a minimum requirement for this to work. But these measurements are also affected by other processes. So they're affected by the cell's microenvironment. There's a large amount of intrinsic noise due to cell cycle variations, for example. And then there's kind of measurement noise, so technical sampling noise that arises from this technology. But the caveat is that these data are very plentiful and very, very high dimensional. So it's not uncommon to hear from researchers in this field who say like, oh, but we have tens of thousands of genes, so there must be enough signal to overcome these limitations. And we wanted to investigate this a bit further. And yeah, I mean, you can take basic systems and just to kind of make the point that actually this is not guaranteed. So even in a simple example where we only have, I think, 4 generations and 20,000 genes, this is not guaranteed. So here I've just shown a very basic example on C. elegans, where kind of even at the 8 cell stage, if you try to use the entire transcriptome, you will not resolve the lineage. You will kind of get some sort of distance in gene expression space, but that will not reflect the lineage in general. You can find some, at least at the 8 cell stage here, some optimal subset of genes that do reflect the lineage, but these are highly variable from batch to batch. And then at the 16 cell stage, even that kind of breaks down. Okay, so I wanted to develop a model for trait evolution to kind of help understand this and maybe just help illustrate the point that this is not guaranteed. So I assume that traits evolve according to an Ornstein-Uhlenbeck process. And the point here is just that we have a kind of developmental signal and a lineage signal, and we want to find out how these interact. And that this is kind of trait dependent, gene dependent, and also cell type dependent. So if we let xai be the value of trait I in cell A, which has some associated cell type at this time, then we can work out the distance between two cells of potentially different types. And then our final observed measurements will, of course, be the combination of all of these differences across all of the types, or sorry, all of the traits. So this is the kind of framework that I'm working in. So I first want to look at the question of resolving an asymmetric lineage. So this is a lineage where we have two different cell types. Here I have an outgroup with the same type as one of these. And, I mean, we can tell immediately that if we don't account for the differences from the rest of the cell type, we have no hope of resolving this triplet here. So the base unit I'm going to work in is triplets. So we have a trait that is dependent on these cell types. So here I've kind of numbered these 1 and 2. And we assume that we have estimates for these kind of mean values for each of the types, which is a big assumption, but this is maybe, this is something that people understand. So I've let epsilon reflect the error in this estimation. And kind of across all these regimes, unless you're in very, very optimistic regimes, maybe down here, the point here is that you have to get really, really good estimates of these mean values in order to resolve the lineage. Otherwise, you're getting completely conflicting signals. So this is, in general, quite a messy thing to do. Okay, but what if we're in an even better case than this? What if we are in either a case where our gene is not developmentally aligned, or we only want to resolve a symmetric lineage, for example? We know that we have a symmetric lineage. Then can we do any better? It would appear so, but it's still a messy problem. So in this case, now that we don't have to deal with types, we can get kind of a bound on the entire tree reconstruction, similarly to what we did before. So this is again using triplets as our kind of base unit, and then creating a bound across all the triplets in the tree. And here are these results in terms of kind of our OU parameters. So we have a signal to noise ratio. So I've just added in Gaussian noise here. And then we also have this kind of selection alpha parameter here. And even in this very, very ideal scenario, there are only a few cases where we can very accurately resolve the tree. And even these are limited to only about kind of 13 or so generations. And these are nice kind of synchronous generations. So if we have any kind of very, very short cell division events, this will almost completely break down. And even here, in this, again, very ideal scenario, we need tens of thousands. So I don't think these results are necessarily surprising, but I hope they do illustrate the point that this is actually maybe expected to work, not expected to work more often than not, and that we should be careful with these these data and kind of better understand how they can actually be used in practice. So yeah, I mean, the point at this stage is just that there's a lot of unknowns, and that we really still need high quality lineage tracing to validate these methods and these other sources of data. And this is something that I'm looking forward to doing next. Unfortunately, it's just kind of a limitation right now that there's not a ton of data sets to work with to actually validate these, but hopefully these are in the pipeline, and hopefully these results are to come soon. And in particular, I'd like to investigate DNA methylation data as these data sets again start to come out and be routinely produced, because this appears particularly promising and has kind of a few less sources of these problematic noise associated to them, but we'll see. I think it's a similar picture. From there, I just want to talk about another project that I've been working on that's related, which is a phylogenetics problem. So once we have the tree, or suppose we have data that reflects the lineage, how do we connect this to the underlying cell population dynamics? This has been kind of fun because typically, or a certain class of phylogenetic methods rely on the birth-death model, but we think this is a really terrible model for cell population dynamics. So we thought about what happens when we relax this assumption. What if we have, for example, a gamma or Erlang distributed lifetimes for our cells? And this allows us to capture a broader range of tree shapes. So here, this is kind of a typical birth-death tree. And then as we increase the shape parameter here of this Erlang distribution, then we're able to capture kind of much more balanced trees and a much more broader class of tree shapes. So this is a project I've been working on with a PhD student, Yulia, here, about implementing this as a tree prior in a popular Bayesian phylogenetic software. Yeah, and applying this to actual data from developmental biology, mostly based on lineage tracing data so far. So in a reanalysis of mouse embryonic stem cell data, we found that this model kind of better captures known features of these published data. So previous estimates that were based on the birth-death model were kind of extremely biased in this case, and we were able to actually better capture the known features. In this case, we had very high quality ground truth data here. Okay, and with that, I would like to thank you. I'd like to thank Tanya and kind of our lab here, especially Yulia, Antoine, and Laura, and all of our lab here at CEVO. Thank you.
I guess so your inference of the lineage is interested in like the precise resolution of the lineage, and I'm curious in the kind of the downstream application of those lineage trees. Like, to what extent is it necessary
to get
every single node right, or is the resolution that you can get sufficient for the downstream purposes?
Yeah, that's a great example. I would say for now, we're definitely interested in relatively small lineages where we do want a very high level of accuracy. And for the theoretical reconstruction guarantees, it's a lot nicer to work with in this framework where we want to get every single triplet, every single split correct, essentially. But yeah, I am interested in relaxing this and how we can tackle this analytically. But yes, so for now, we're interested in exact reconstruction, but for sure, that's a bit strict.
Yes, I guess I was sort of curious with your OU simulations. Do you get a sense of sort of intuitively what's breaking down there? Why the trees aren't reconstructible in that case?
Yeah, we're just getting conflicting signal from the cell state, and that completely corrupts the lineage signal. So if we have a purely Brownian process, we tend to be able to reconstruct at least 8, 9, 10 generations fairly well in this. When we have tens of thousands of data points, there's still enough signal there. But yeah, as soon as we have this conflicting signal that's coming from the cell state or the developmental trajectories, then this completely breaks down. And of course, this is very sensitive to all these parameters, but I guess the point I want to make now is simply that this is very sensitive to all these parameters. We don't know, and I don't think the default assumption should be that this works.
I guess I was curious to ask whether we expect epigenetics to be better than, or whether it suffers from the same issue that you're finding here with the expression.
I think it does, but I think ideally it's a bit less noisy, and maybe we can correct for these factors a bit more. I didn't kind of really get into too much detail about how to actually correct for this, because that's kind of another can of worms, I think, but yeah, for sure.
So sort of building on what you were saying, and you had mentioned that DNA methylation had some promise, but when you talked about why the barcodes were great because they're irreversible, but DNA methylation like sort of decays. So it has a time signal that's lost. So it has lineage signal, but it's being lost.
Yeah.
And it seems like in your model, you could include that of whether you had a marker that, loses lineage signal. Have you been able to sort of think about that? Because I think methylation will probably have that issue.
Yeah, I think, and a lot of this comes down to the fact that we are looking at very small scale lineages. So yeah, on the order of kind of 10 generations, where I tend to kind of ignore some of these factors. But yeah, for sure, there's a lot more that we can and should eventually include.